Okay, we'll make a start then. So, firstly, just introduce myself. I'm Mark Ormerod, Deputy Vice-Chancellor and Provost, which many of you will know. And normally, this job is done by the Executive Dean of the faculty, but the Executive Dean of the faculty is in Australia having a very nice time. And we're all going to have a really nice time too, just want to emphasise that. But so I'm doing this as well as the presentation at the end. But it is a really great pleasure to welcome academic colleagues, research colleagues to this lecture from Pip. It's also a really great pleasure to welcome Pip's parents who've come from Australia, so there's no pressure on giving a good lecture at all. It's quite a long way back afterwards. And also Pip's partner, Paul. And I think everyone knows the format of an inaugural lecture, but it's an opportunity for a new Keele professor or relatively new Keele professor to talk about their area of expertise, but also to showcase Keele's research and the discipline that they're in. So I'm just going to say a little bit about Pip's background, which I think will be familiar to a good number of you. Pip's a veterinarian specialising in the study of viruses, and she did her undergraduate degree. I have deleted the bit that said it was a long time ago, but she wrote that, not me. She did her veterinary degree in Sydney University in Australia, and then moved to the UK to work in a mixed veterinary practice in North Yorkshire before undertaking a PhD at Edinburgh University. And that marked the beginning of a love of Edinburgh and also the Scottish rugby team, who regularly test their patience in their ability to snatch defeat from the jaws of victory. Now, I'm from Yorkshire, so I'm struggling a bit with the former in terms of leaving Yorkshire to go to Edinburgh, but as a lifelong Leeds United fan, I can relate to the latter very, very well. Pip then moved to the US, to Cornell University, and you can start to see the track record of some really quite impressive universities that Pip's worked at. So in New York State for 3 years, where she started her real research in terms of studying viruses. And she was then awarded a really prestigious Wellcome Trust Fellowship to return to the UK and spent 3 years at Imperial College researching poxviruses, and has really been studying poxviruses ever since. Pip then spent a year working for the Mongolian government, and I think we'll all be keen to hear a little bit more about that in her lecture, before starting her own poxvirus research laboratory at Edinburgh in 2007, working at the Roslin Institute, where she met her partner, Paul. Pip combined her poxvirus research with specialist training in veterinary pathology at the Royal Dick School of Veterinary Science in Edinburgh, and spent some really happy years there, kind of leading both the research team and working as a pathologist within the vet school. And then, again, I'm reading to something that Pip wrote, but you can sort of get a sense that Pip has moved quite frequently at about a 5 or 6 year basis. I would emphasise that movement has now stopped, and she will not be moving anywhere soon. And in 2016, she moved to the Pip Beard Institute in Surrey, taking up the role of a specialist in, and I may get the pronunciation wrong, Capillary poxviruses, which are a particular group of poxviruses that cause severe disease in livestock. And while she was in this role, there was an epidemic of lumpy skin disease in cattle caused by said virus. And so Pip's years at Pip Beard were very eventful, and she acted as a specialist scientific advisor on a veterinary virology epidemic, while the world was in the middle of a human virology pandemic. And so clearly a really challenging and important time. After 6 years at Pip Beard, the role of Head of School of Life Sciences at Keele was advertised, and I would say I'm incredibly pleased that Pip applied for the role, coming from slightly out of the sector, and it was it was fantastic when Pip came through the field to be able to offer her the role, and even more fantastic that she accepted the role. And in the time she's been Head of School of Life Sciences, she has made a huge impression in terms of her leadership contribution in what is a really thriving and expanding School of Life Sciences, with some tremendous appointments all the way from early career to Professor and Head of School. So it really is a great pleasure to formally welcome Pip Beard, Head of the School of Life Sciences and Professor of Viral Pathology, to present her inaugural lecture, How Did Poxviruses Cause Disease? A Story of Cattle, Lumps, Insects and Leadership. So, Pip.
Well, thank you very much, everyone, and thank you, Mark, for those lovely words. It's a pleasure to have the opportunity to speak to you today about my career and some of the things I've learnt along the way. So I grew up in Australia, in Sydney. This is the family, so Mum and Dad, who look exactly the same as they did 35 years ago. This is my older brother, Rob, younger brother Tom, and sister Suze. And absolutely, it was an idyllic childhood. Just imagine the most beautiful childhood growing up in Australia, and that's pretty much what it was like. So lots and lots of beach, lots of swimming pool, loads of cricket, both at the Sydney Cricket Ground and in the back garden, holidays in the Blue Mountains, Harbour Bridge, Opera House, all that kind of stuff. It was really amazing. Between the ages of 11 and 15, I went to James Roos Agricultural High School, which is a specialist high school in the northwest of Sydney. And as you can tell, it specialises in agriculture. And that was a really great fit for me because of my ambition to be a vet. So I've always wanted to be a vet. And I even remember now when I was about 15, we did a microbes module in high school, and we learnt what tetanus was and why it's called lockjaw. And I remember thinking, this is amazing. This is just the coolest thing I've ever. And I was looking around at all my friends thinking, don't they realise this is so amazing? And I think that love of microbes, of viruses, bacteria, things like that has never really left me. As well as discovering my love of microbes, I also realised when I was in high school that I am definitely really driven by knowledge acquisition. I just love learning new things. Every day I learn something new is a good day. So I really enjoyed high school because of this, because it's all about learning new things. At the end of 6 years, I got the academic qualifications required to go to vet school. So I went off to Sydney University. And of course, doing a veterinary degree, it's all about knowledge acquisition and understanding concepts. So I had a very rewarding time at Sydney University. I did some small animal work, but actually what I really loved more than anything, I'll just move that pointer, was actually the large animal work. So these are some Angus cattle in a farm where I was doing some practice. This is on the Central West Slopes and Plains. And then a flock of merino sheep. There are many merino sheep in Australia, and these are on the Central Tablelands. While I was doing my veterinary degree, I had the opportunity to go to Cornell University just for 10 weeks one summer as part of their leadership program for veterinary students. This is a program where Cornell offers this opportunity of 10 weeks at Cornell to 24 vet students from around the world, and then they bring them together and they give them 10 weeks in a research lab. Now, at that time, a research career really wasn't on my radar, did not know one end of a pipette from the other, but it was a 10-week, all expenses paid holiday in the US, so obviously I was going to go. And I was put in the lab of Professor Joel Baines, who is a specialist in herpes virus. So herpes viruses cause herpes simplex virus. And I actually really loved it. I definitely fell in love with research during these 10 weeks. And I remember even now in a dark room developing an X-ray during this summer holidays. And it was in the olden days where you had the developer in a tray with your radiograph in it and you were joggling around and watching those bands develop. And I found a result that showed me that the UL28 protein of herpes viruses binds to the genome of the virus. And I suddenly realised I was the only person in the world that knew this because I had just discovered it. So nobody else knew this fact, and I still remember how exciting I found that. So it wasn't just that I really enjoyed that knowledge acquisition stage, but I was actually now generating knowledge, and I really loved that as well. Not just knowledge acquisition, but actually knowledge generation. Now that's to remind me to say, Joel was a fantastic mentor. I'm still in touch with him now. He's been a constant mentor throughout my career. He's also a veterinarian, but he does work on fundamental virology, and he showed me that vets can have many different career paths. You don't need to do research just on veterinary topics because you're a vet. You can be a researcher on many different things. He is one of the smartest people I've ever met, and he's one of the best herpes virologists as well. Despite the fun I had in Cornell, when I finished my vet degree, I still really just wanted to be a vet. So I moved to North Yorkshire, to Malton. It's a town sort of in between Scarborough and York. And I went into mixed animal practice. It's a lovely, lovely part of the world, a very traditional market town, and I love being part of that traditional farming community. I learned a lot about very different sheep. These are black-faced sheep, very different from merino sheep, so there's lots of different animal husbandry things to learn. But I then was offered the opportunity to do a PhD in Edinburgh at the Mordun Research Institute, which was associated with Edinburgh University. And Mordun had a really good reputation for doing research into infectious diseases of livestock, and that really fitted my interests. So I moved up to Edinburgh. The title of my PhD was The Epidemiology and Pathology of Paratuberculosis. Paratuberculosis is a bacterial disease. It's caused by infection with Mycobacterium avium subspecies paratuberculosis, and it causes disease in ruminants. It causes a chronic infection of the intestine, which results in diarrhea and weight loss. Example there. Now, when I started my PhD, the dogma was that this bacterium only caused disease in livestock. It only infected ruminants. So it had a very narrow host range. And my PhD was spent trying to figure out whether this was true. Does this bacteria only cause disease in ruminants, or does it actually have a wider host range? So I spent 3 years driving around country Scotland picking up dead wildlife from farms that had a paratuberculosis problem. And I found out that actually they could be infected by this Mycobacterium species. I found evidence of paratuberculosis in rabbits and pretty much everything that eats rabbits, such as weasels, stoats, crows, jackdaws, all of these kinds of animals as well, and foxes. And this is a histopathology picture of the lymph node of one of those foxes. And the little red things that you can see are actually this Mycobacterium in the lymph node of the fox. So I could then let the farmers know that actually you might need to think about controlling your wildlife if you want to control paratuberculosis. Paratuberculosis is a really difficult disease to eradicate once you've got it on the farm. It's a really long-term problem. So the farmers were then able to make some changes. They made sure that their feed bins were rabbit-proof, so the rabbits couldn't get in there and potentially contaminate them with this bacteria. And so that was, again, sort of another stage along. Not only was I discovering that new knowledge, but I was actually then giving it to people who could make differences, make changes, actually act on that knowledge and make a difference. And that's to remind me that my supervisors, Dave Buxton, who I couldn't find a picture of, but I'm really sorry about that, because again, I'm still in touch with Dave, and Susan Rind, who is a professor at Edinburgh University now. Fantastic supervisors, could not have been more supportive, and actually both vets as well, both vets that really loved research. So again, 2 really strong role models for me. They're vets, and Susan being a woman as well, being very successful in research, and that was really important at that early stage of my career. So once I'd finished my PhD, I then went back to Cornell to work with Joel again. And I was a little bit worried because my PhD had been quite vetty. I mean, driving around doing postmortems on lots of dead animals. Whereas what Joel does is really hardcore molecular virology. There's no animals involved in this at all. It's really fundamental kind of stuff. And I was thinking, gosh, I wonder if I would really enjoy this for a full 3 years. And I did. It was absolutely fascinating. I was looking at this little complex here called the terminase complex, which is encoded by the virus, and it latches on to the genome of the herpes virus, which is in this great big long strand, and it brings it to this preformed capsid. And then it recognizes the capsid, docks, and then rams the genome into the inside of that hollow capsid. And it does it so tightly, it's almost at liquid. It's the most amazing little molecular motor. And so my role was looking at what this little terminase enzyme was made up of and how it worked. Again, it was really fascinating, really interesting, really helped by good mentorship and support from Joel. He was a great line manager and mentor. But again, really exciting times. And I was then awarded the Wellcome Trust Clinical Fellowship to work at Imperial College in London in the lab of Jeff Smith. And this is where the poxvirus bit starts. So that's the lump part of the title, because poxviruses cause lumps, cutaneous nodules on the skin. So I started, again, taking that molecular virology, but doing it with poxviruses. So how do poxviruses interact with the cell on a molecular basis? How does it cause disease? What does it actually do? So I spent 4 years working on that at Imperial College. And in parallel with that, I also did a diploma of management with the Chartered Management Institute. And this was sort of 8 days scattered throughout a year, and Imperial paid for it, bless them. And that was brilliant. It was almost like sort of management and leadership 101. So I learned about how to do annual appraisals, how to recruit people effectively and fairly, how to deal with budgets, what different leadership types look like and how to recognize those in yourself. All this really fundamental stuff. I still use the stuff that I learnt on that management course now, and it was so useful to have it when I was really just setting up my own lab, to have that fundamental. So that's the reason why I keep encouraging everyone to do these leadership courses. It's because I found it was really important. And then I took the year out to go and work in Mongolia. So I was a specialist veterinary advisor to the Mongolian government. So I lived for a year in Ulaanbaatar, which is the capital city just here. Has anyone been to Mongolia? Anyone in the audience been to Mongolia? Oh, it is the most beautiful country. It's a bit of an adventure destination, but really advise you'll never regret going. It's really the most fantastic place. So I was sponsored by the Wellcome Trust when I was over there to do some work on poxviruses of livestock in Mongolia. And I was sponsored by the Royal College of Veterinary Surgeons to do some audits of their regional veterinary laboratories. So that involved me going on these 2-week trips into the countryside. So the first one I did was out to the east. So we went down to the Chinese border and up through Sukhbata, through Dornod, and back through Hinti to Ulaanbaatar. And we would stop at about 6 or 7 different places, little towns, and then go to their veterinary laboratory and have a look at what they were doing, and what they were doing well, what they were doing badly, what did they need help with. And then the second one, we went out west. We went all the way out to Berlanga, Ulustai, and back by Tetelig to Ulaanbaatar. And then the final one up north to the Russian border through Dakhan. It was amazing, just the most amazing experience. Mongolia, it's a really unique society that is still fundamentally based on a type of nomadic herding system that has not changed in 1,000 years. So there's fewer and fewer, but still a substantial portion of the Mongolian population live in this nomadic lifestyle. So they live in these white tents, you can just see in the back here, called gers. And then they herd mixed sheep-goat flocks, like you can see here, and they still do it mainly on horseback as well. And this is a revered kind of lifestyle. Everybody wants to be able to have this lifestyle, and it still forms a really fundamental part of what Mongolia is, what all of their culture is based on. These are the colleagues that I worked with while I was in Mongolia. So this is me up the back. And these are all the colleagues. And that's at the State Central Veterinary Laboratory, which is the big veterinary laboratory in Ulaanbaatar, which is the main veterinary laboratory. I did some capacity building work while I was there. This is my colleague Garelma, and we're doing a diagnostic PCR. And my colleague Dashka, where we're doing some cell culture. I don't usually wear this much PPE for cell culture, but it was a really, really dusty environment in Ulaanbaatar, so we put a little bit more PPE on because we had a bit of a contamination issue. We also did some research into sheeppox and goatpox. These are 2 important poxvirus diseases of livestock. There hadn't been any sheeppox or goatpox in Mongolia for about 20 years, but then there was an outbreak of sheeppox in 2006, and then an outbreak of goatpox in 2008. And 2008 was the year that I was there. So we did some research into best way to diagnose, most appropriate and effective control programs. But we also did a little bit of phylogenetic analysis on the viruses that caused these outbreaks. And we were able to say that the most likely route of entry into Mongolia was probably from China through illegal trafficking of livestock. So once I'd done my year in Mongolia, I then went to Edinburgh to take up a permanent lectureship. And I was there for 7 years, and a lot of the research that I did there was done by 2 amazing postdocs, Dr. Isma Haga and Dr. Tali Pachek-Jowers. And this is Kate Harrison, who was my first PhD student. So we called ourselves Pox World. This is a great logo that was drawn for me by Dr. Charlotte Cook, who's one of my later PhD students. And I kind of went back to that molecular virology that I loved so much. So looking again at poxviruses, we used the prototype poxvirus, vaccinia virus, and I did this high throughput screen. So I used siRNA technology, which was really cool and trendy back then, to knock down individual proteins within cells. So 6,000 different human proteins, knock them down with siRNA technology, and then put the virus in, and to see which of these human proteins was either proviral or antiviral. Did the virus replicate better or worse in the absence of this protein? And this was a really good project to do as an early career researcher. High throughput, absolutely tons of data, loads of really, really interesting hits that were all very novel, and I could take each individual hit, do a bit more characterization, and then get a lovely paper out. So it was, definitely helped by having two amazing postdocs, but it was a really successful sort of start to the career. In parallel with leading Poxworld and leading this program of research, I also did a residency in anatomic pathology, and I became an associate of the Royal College of Pathologists, and then 1 week in 4 I would work as a diagnostic veterinary pathologist in the vet school up there. And this was kind of combining the best of both worlds. I was getting to do my lovely molecular virology with all the Poxworld stuff that I was doing, but at the same time, I was able to really ask, why do livestock get diseases? So I specialised in the livestock for the pathology. What causes disease in livestock? There would be a different thing coming into the post-mortem room every day. It was a lot of fun. And the other thing I did in Edinburgh was meet Paul. This is the lovely Paul. And this is a holiday we had up in the Southern Highlands. This is just above Comrie, Loch Lednock. It's really beautiful. So after I'd been in Edinburgh for about 7 years, I was asked by the Pirbright Institute if I would be interested in coming down to work for them to lead their veterinary poxvirus research program. The Pirbright Institute is a bit unusual. It has a very, very narrow focus. It's in Surrey on the outskirts of London, and it has a very narrow focus. So it only really carries out research into viruses of animals, so veterinary virology. And it has a particular focus in exotic diseases. So it's a high containment institute. It works at containment level 4. So this is the containment level 4 laboratory or building at Pirbright, and the Poxworld from 2016 to 2022, we were in the third window from the end. That was the poxvirus laboratory just there. And the most important poxviruses of animals by far are the capripoxviruses. This is the genus of poxvirus that's made up of 3 species: sheep poxvirus, goat poxvirus, and lumpy skin disease virus. All of them cause severe systemic disease in ruminants, so sheep poxvirus in sheep, goat poxvirus in goats, and lumpy skin disease in cattle. It's kind of like smallpox in ruminants is a good way of thinking it. So multiple cutaneous nodules, internal lesions, really nasty diseases. And back in 2016, when I took the job, I was also asked to take on the role of expert for the World Organization for Animal Health, which is kind of like the animal equivalent of the WHO. So they needed someone to be the capripoxvirus expert for WOA. And I said, I could probably do it, because I did do some work with goat pox and sheep pox when I was in Mongolia, but I didn't know a ton about the capripox viruses. And the person was saying, don't worry about it, it's not going to be that busy. Yeah, there's really not much going on in the capripox virus world, so you'll have a really quiet time. And literally the next week, there was an outbreak of lumpy skin disease in Europe for the first time, and it was not a very quiet time at all. So lumpy skin disease, as I said, it causes disease only in cattle. It has a morbidity of 10%, mortality of 1%. So if you imagine you have a flock of 100 cattle, if you have an outbreak of lumpy skin disease, 10 of those cattle are going to get sick and develop those lumps, and 1 of them is going to die. One of the interesting things about lumpy skin disease virus is it's transmitted by insects. So it is vectored by insects biting insects, hematogenous insects. And that's a calf you can see with lumpy skin disease covered in these large necrotic lumps. On the right-hand side, That shows you why lumpy skin disease is so important at the moment. So in grey are the countries that reported their first case of lumpy skin disease before 2012. And then in green are the countries that reported their first case of lumpy skin disease between 2012 and 2018. And then in 2019 and onwards, those are the countries in red. So you can see that it had been localized just to Africa. There hadn't been any reports of lumpy skin disease any further. And there seemed to be, again, this dogma that lumpy skin disease wasn't able to occur outside of Africa. There was some fundamental thing that was missing, and we would never get lumpy skin disease apart from in Africa. You can see this is epically wrong. So the outbreak started in the Middle East. It came up through Turkey, came into the southeast of Europe, so in the Balkans, Greece and Bulgaria. And then it spread really rapidly across Russia, and then really rapidly across Asia, and it's now threatening the northern part of Australia. So yeah, it was definitely a very busy time. The thing about lumpy skin disease is it's also a neglected pathogen. Because it had only occurred in Africa for so long, there were huge gaps in our knowledge. Very little research had been undertaken into lumpy skin disease. One of the key knowledge gaps was how is it transmitted? So during this big outbreak, tens of thousands of cattle died. It cost countries millions of pounds. A lot of people were saying to us, well, how is it transmitted? How can we block that transmission? And we just kept saying, we really don't know. So that's what we focused on in Poxworld, looking at the transmission of lumpy skin disease virus. So I mentioned that it's transmitted just by insects. You can't transmit it directly. If you have a cow that has raging lumpy skin disease and you put it in the same small room as a naive cow and leave them there for a week, that naive cow will not get infected. It won't even seroconvert. So there is no direct transmission of lumpy skin disease virus. I split donor up into clinical and non-clinical because you remember I said that only 10% of animals in that herd will actually get clinical disease, so only 10 of them will become lumpy. And so we don't know what impact that plays in the transmission of the virus. And then we looked at acquisition, retention of the virus in the insect, and then deposition of the virus into a naive animal. The key questions that people were asking us were, what vector species is it that transmits lumpy skin disease virus? Is it like malaria and plasmodium, where there's just one mosquito type that will transmit it? Does it have a really narrow vector range? Or do tons of different insects have the capacity to transmit? We wanted to know how long the insects retained the virus for, because that would be really important in telling us about how quickly they could transmit it. If an insect can only keep the virus there for 30 minutes, well, it's not going to get far in 30 minutes. But if the insect's able to retain that virus for 3 days, it can travel a long distance in 3 days. So that was a key piece of information we needed to know. We needed to know the role of these non-clinical cattle. Were they actually important in the transmission of the disease, or was it just the clinical cattle? But the really important million-dollar question was, how likely are these different species to actually transmit the virus? When we started this work, just 2 papers were published. One showed that flies could not transmit lumpy skin disease virus, and one showed that mosquitoes could. But what we wanted was quantitative data. We didn't just want that yes, no answer. We actually wanted to know how likely was it to happen, because that then really enables you to put in or design a much better control program, a proportionate control program, which you are pretty sure is going to be effective. So we developed an experimental model of lumpy skin disease. Actually, did I say that? These are the guys in Poxworld who were involved in this study, and I know some of them are online. Hi, guys. So we developed an experimental model of lumpy skin disease virus. This is one of my colleagues, and you can see in her hand here, she has a pot of insects, and it's a pot, and it's in a cardboard box, and it has gauze over the top. And then you take it and place it upside down on the animal, and it's able to feed on the cow. So we would feed these insects on clinical donor animal, and then we would take them to the recipient animal and let them feed on that recipient animal. And most importantly was that quantitative bit I was talking about. So we would then take each individual insect and take them back to the lab and look to see, had they actually fed on the donor? Had they acquired the virus from the donor, and had they then fed on the recipient? And in that way, we were able to actually get this quantitative data to tell us how likely it was that each of these different insect species could transmit the virus. So we looked at mosquitoes, Aedes aegypti. We looked at biting flies, Stomoxys calcitrans, And we looked at little midges as well, Culicoides nabeculosis. And we were able to come up with things like it takes 8 virus-positive mosquitoes to transmit lumpy skin disease virus. I was saying that it's impossible to get that direct transmission. It is very easy to get insect-borne transmission. These insects are super good at transmitting lumpy skin disease virus. You only need 8 positive mosquitoes to get that transmission to happen. So this kind of summarizes 6 years' worth of work in one slide. Which vector species can transmit? Multiple different vector species can. So it is not one of these very specific pathogens. Pretty much anything will bite a cow, will pick up the virus, and potentially be able to transmit it. How long do they retain the virus for? A long time. So we looked up to 8 days, and potentially it could be even longer. 8 days is a really long time for this type of transmission. And you can imagine mosquitoes, and particularly flies, they can go a long way under the right conditions in 8 days. We definitely found that clinical cattle were by far the most important ones as drivers of the transmission of the virus. It was almost impossible for these insects to pick up the virus from non-clinical animals. We looked at 1,021 insects that had fed on these non-clinical cattle, and only 7 of them had acquired the virus. So compared to the clinical animals, where almost every insect acquired the virus from these nodules, they're absolutely hooching with virus. So that was a really important finding as well. It's all about those clinical cattle. And finally, how likely are these different vectors to transmit? Both the flies and the mosquitoes definitely can, but when you actually calculate it, it's all about those Stomoxys calcitrans biting flies. They were 5 times more likely to transmit than the mosquitoes. So if we hadn't done this quantitative thing, we wouldn't have been able to find that out. We would have just seen transmission by at least 2 different species. But because we could do it quantitatively, we could say the flies are by far the more likely ones to transmit. So then we could take this information to the stakeholders, to the people who really needed to know this. We could tell people that vector control is very challenging to control lumpy skin disease virus. In the start of that epidemic in Southeast Europe, some countries were trying a control measure which involved flying aeroplanes full of pesticide across great swathes of the countryside and trying to kill all vector species. So we were then able to say that is not going to work at all. It's going to have huge environmental harms, but also you're going to have to kill pretty much every flying insect, and that's just not possible. So we really recommend that that's not a control measure that we use. We were able to say, focus on the clinical cattle. One of the early control measures that they used was a complete stamping out of all cows in a herd that had lumpy skin disease. So you can imagine you've got a herd of 100 highly prized dairy cattle. 10 of them have got lumps, and you have to kill everything. And we were then able to say, look, you probably don't need to kill those 90 that are unaffected. Just get rid of those lumpy ones as quickly as possible because they're the drivers of the transmission of the disease. And the non-clinical ones, you are probably fine just vaccinating them to be sure, but then you don't need to kill them. So this made a big difference in the take-up of these control measures. A lot of farmers were much happier just killing the clinical ones than having to kill everything. And we had that quantitative data, which people can now use to put into their own mathematical model, which has their own particular ecological values in. But we were able to highlight how important biting flies were. So this is a buffalo fly, which is really common in northern Australia. And you remember Northern Australia is the next place that's going to get lumpy skin disease. And it's a ferocious feeder. It feeds about 50 times a day on many different animal species. So we were able to tell the guys in Australia, for your lumpy skin disease control, you really need to take care of this buffalo fly. It's going to be a super good transmitter of lumpy skin disease virus. So that was the transmission side. The other big topic we looked at when I was in Pirbright was making a better vaccine for lumpy skin disease. And this work was carried out by these ex-members of Pox World. And this was part of the Defend project. So this was a really large 5-year, €5.6 million consortium that was funded by the EU as part of the Horizon 2020 framework. And I led the work package on developing a better vaccine for lumpy skin disease, but I also led the overall consortium as well. And this is the consortium here. There were 30 different partners, government, industry, academic partners from 16 different countries. So it was my first experience of that kind of big science group together, teamwork thing, and it was super effective. This is definitely the way to make big breakthroughs really quickly. Just get a load of different people who all have different specialities, put them all together in a big consortium, and it is incredibly effective. So, yeah, a really positive experience. Now, while I was at Edinburgh and at Pirbright, I kept working on leadership skills because I think they're really important. So being the lead of PoxWorld and then being the lead of Defend were really great leadership experiences. Very different. PoxWorld was very traditional research group, whereas Defend, it was much more kind of matrix management. I wasn't direct line manager of anyone in that particular consortium, but I still needed to get them to work together as a team and persuade them to do things. I also continued formal training, and I can highly recommend the Windsor Leadership Trust. I've done some really good courses with them on leadership. They're 3 days, a week long, really effective. I've also kept up with the literature around leadership. It's very accessible, very readable, very interesting. And I would definitely recommend this book, Through the Labyrinth, which is by Alice Eagley, who's an academic from Chicago. She's probably retired now. It's a really good book. It's all about women in leadership. Very evidence-based, very academically written. It's really good, really powerful. And the other thing I did was keep up my mentorship as well. So I mentioned Susan and Dave Buxton and Joel Baines and Jeff Smith, and I'm still in touch with them now. I still ask them for advice and support, and that kind of mentorship is really invaluable. And now I'm here at Keele. So I have taken a little bit of time away from having that active research lab, so PoxWorld is kind of in mothballs for a little bit while I concentrate on leading the School of Life Sciences. So that balance between leadership and research is probably a bit more in that kind of direction at the moment. It has been an absolute honour and privilege to lead. I can see many of you in the audience. It has been absolutely brilliant to have the opportunity to lead you guys, particularly in such an exciting time. Our student numbers are going up. Our research is absolutely flying. But most of all, you guys really believe in what we're doing. The motto is definitely what we do in the School of Life Sciences. We provide high-quality, innovative education to students from all backgrounds, and we do life science research, which provides benefits to people, communities, and the society. So that's all really important as well. So a massive thank you to all. As, oh my goodness, I learnt what sprees and prees were, and shrug and shrek and feck and all the different things. You guys have been amazingly patient with me. So final slide, just to sort of sum it all up. As I was doing this lecture, I thought that actually this kind of knowledge generation thing is really important to me. It really is a driver for me. I love the knowledge acquisition, learning what tetanus is. I love the knowledge generation, and that's what I've tried to do through my research. And then the knowledge sharing is also really important as well, telling people about the things that you've discovered so they can actually put them into practice. And that kind of leads me on to the next point, the expert voice. So all of us as scientists are super specialists in one particular area. I remember when I was going for my PhD viva, and someone said to me, "You know more than anyone else in the world about the role of wildlife in the epidemiology of paratuberculosis. No one knows more than you." And they were right. I can see it now. They were right. And that's true of everyone, maybe not my parents, but all the scientists in the audience. We have one particular thing that we are super specialized in. And there will be 1 or 2 times in your career, this goes for scholarship as well as for research, there will be 1 or 2 times in your career where actually that's incredibly important information. And it's important that we speak up at that point. And I had to do it during the lumpy skin epidemic, and I also did it a bit during the monkeypox, or the mpox outbreak in 2022. It's not something I particularly enjoy, it's not something I'm natural at, and I definitely feel a real weight of responsibility, and definitely some parts of the mpox was just downright unpleasant, but it's still important for us to do that, because I find it very frustrating when you see non-experts, scientists, but non-experts, non-scientists, who put their voices forward, especially on social media. And it is our obligation as specialist scientists to speak up when it is our turn to do it. It's kind of like leadership in your focused area. And that leads me on to the sort of leadership as a more global level. One of the things I've found very worrying recently is the increasing global fragmentation that we're facing, the increasing fractures that we're seeing in society, more of them, and they're becoming wider. And a lot of this is driven by the rise of populism and left-wing and right-wing extremism, but it's really characterized by the misuse of truth and the misuse of knowledge, usually by very high-profile people. And I find that really annoying because this knowledge and it is such an important thing for me. I value it very highly. And I was thinking universities are super important countermeasures to this. We have a really consistent sort of design and structure, and we also have some very clear principles around this. So I've been to universities all over the world, and they all look pretty similar. They all have lectures, they all have academics, they all have students, and the students learn and they do exams. It's a very consistent model globally. So you have this consistent feature which you then put together with the principles of universities. We are all about discovering new knowledge and then disseminating it for the use of everybody and for uncovering evidence that can then be used to make good decisions. And this is incredibly important balance when we're in a world with all this, what is the truth? What is the misuse of truth? And I think we should probably speak up about this more often. Universities are getting a kicking regularly in the media, and this is one incredibly important thing that we do, and we'll do more often over the next few years as this voice of reason against this fragmentation that we're seeing at the moment. So I would encourage people to take on that more global sector-wide leadership role and to speak up about this as well. And that's it. A massive thank you to everyone from Poxworld. These are all the people who've worked in Poxworld over the years, the funders as well. A big thank you to Paul, big thank you to all the family, to Tom, Suze, Rob, Kate, Soph, Jess, Miles, Jobs, and Mum and Dad. The fact that they've come all the way from Australia is emblematic of the support they've given me throughout my career. No matter what I said, they'd always be like, that sounds interesting. This is Poxworld here, my colleagues in Mongolia and the Defend Consortium, and all of you for listening. Thank you very much.